Hey guys, in this video, I'm going to explain to you the formation of action potential in the neuron. Human's body cell is not simply just neutral. In fact, the inside of the cell is having negatively charged, while the outside of the cell is having positively charged. This electrical charge difference across the plasma membrane is known as membrane potential. The membrane potential is having the reading of negative 70 millivolt. You may be curious, what factor contributes to the formation of membrane potential? There are three factors. The first is the presence of fixed end ion in the cytoplasm. Fixed end ion refers to those protein and DNA molecule which is negatively charged. The presence of negatively charged protein and DNA molecule in the cytoplasm makes the inside of the cell become more negative than outside. The second factor is the sodium-potassium pump. Sodium-potassium pump actively transport 3 sodium ion out of the cell and 2 potassium ion into the cell. This makes the cell to lose 1 positive charge in each transfer. The third factor is the potassium channel and the sodium channel. Potassium channel transport potassium ion out of the cell, while sodium channel transports sodium ion into the cell. However, the plasma membrane is having more numbers of potassium channel than sodium channels, which means the cell will be losing more positive charge than gaining the positively charged ion. All these three factors together contributes to the formation of membrane potential. Now you understand about membrane potential. What about an action potential? Action potential is the technical term for impulse. Action potential is defined as a short-term change in the electrical potential on the membrane surface of a neuron or muscle cell in response to stimulation, and then this leads to the transmission of an electrical impulse that travels across the cell membrane. Which means when the cell receives a stimulus, this stimulus will create a short-term, temporary change on the membrane potential. This short-term, temporary change on the membrane potential referred to as action potential or impulse. Let's look at the step how to generate an action potential. The diagram here showing part of the axon in the neuron. In the neuron, they are having voltage-gated sodium channel which transports sodium ion into the axon, and voltage-gated potassium channel which transport potassium ion out of the axon. In the resting stage, when the neuron did not receive any stimulus, both voltage-gated sodium and potassium channel remain closed. Inside the axon, it is having high concentration of potassium ion, while outside the axon, it is having high concentration of sodium ion. When the neuron did not receive any stimulus, the neuron is remaining at its resting stage. At this point, the inside of the axon is negative, outside is positive, same like normal body cell. The axon membrane is having the membrane potential reading of negative 70 millivolt. When you receive a stimulus, for example, you push your coffee, this creates a stimulus which opens some of the voltage-gated sodium channel. Sodium ion is allowed to enter into the axon. The entry of sodium ion makes the inside of the axon become less negative. However, at this point, action potential or impulse is yet to be generated. To generate action potential, you need to have sufficient number of sodium ion to enter into the axon to make the inside of the axon become less negative. When the number of sodium ion enter into the axon is sufficient, and this will make the axon with membrane potential reading increase to negative 50 millivolt. When it reaches negative 50 millivolt, the membrane potential is at its condition of threshold stage. When it reaches threshold stage, all the voltage-gated sodium channel will open. Large number of sodium ion will rush into the axon. At this point, action potential or impulse is generated. This makes the inside of the axon become more positive than outside. And now the axon is at its depolarization stage. Depolarization is referring to its reversing the original polarity of the axon membrane. When the membrane potential reaches positive 40 millivolt, all the voltage-gated sodium channel will close, and now it moves to fourth stage called repolarization. At repolarization stage, the voltage-gated potassium channel will open. Large number of potassium ions were moving out of the axon, and this makes the inside of the axon returning to its normal scenario, which is inside negative, outside positive. However, the voltage-gated potassium channel closed slowly. This causes the axon to lose excessive amount of potassium ion, and this makes the inside of the axon become extremely negative than its normal condition. This is referring to as hyperpolarization stage. However, after some time, the voltage-gated potassium channel closed completely. The membrane potential will reset to its normal condition, which is negative 70 mV. This marks the end of the generation of an action potential.